Welcome back, everyone. We are still continuing with this series, the Life Sciences series. So we're still in term one. So for those of you who are new here, I'm Andrew Command and I make videos on the CAPS curriculum. So that's Maths Lit, Geography, Life Sciences, and of course, Physical Sciences as well. Now for the requests, guys, I'm still waiting. So if you have any request, any topic that you feel like we can start with, or maybe, of course, break it down before we venture into other terms, or maybe it's a topic from another term. Actually, yeah, we can do that as well. So we don't have to go by, of course, the curriculum. Okay. So let me know down in the comments and share, guys. Let's share this channel so that it can grow. Okay. So in the previous video, we were looking at DNA replication, looked at how DNA replication occurs, described the process, and of course, I used diagrams, and I think it's very much beneficial and much easier to understand concepts when using diagrams. So if you don't know that video, please check it out. I'll leave it in the description and it should appear above on your right. Okay, so let's look at our CAPS document first. So this part, the DNA part, the DNA section part, we have already done it. Okay, so we are done with that. We've covered everything that needs to be covered here. Again, guys, if you do not or you did not access these videos, please start with them because we're about to get into RNA now. Okay, so if you really remember clearly, okay, so for DNA, we said that both of these are actually nucleic acids. And we said nucleic acids are a type of organic compounds, right? Organic compounds, we said those, they are carbon, they are made by carbon, most importantly, okay? So we said that these two nucleic acids, they are made up, of course, of nucleotides. Okay, so both of them are made up of nucleotides. I even made an example of a wall. So when you have a wall, what you see is a wall, but actually a wall is made up of bricks, right? So bricks in that case are the building blocks of that particular wall, right? So in this case, that means the wall in this analogy is the nucleic acid, and then these bricks, these single bricks are your nucleotides. They make it up. Okay, so again, if you really remember clearly, we said that DNA has a unique structure which we call a double helix. Okay, we said double we said double because it has two strands, and then we said helix because these two strands, sorry about that, these two strands actually look like a twisted ladder like that. Okay, so that is for the helix part. Okay, the twisted ladder. The double, of course, means it is double stranded. It has two strands that are connected together. Okay, so and then we also described these nucleotides of DNA, particularly DNA, which we said that it is made up, of course, three elements, okay, or three parts, which is the deoxyribose sugar. Okay, so note the name of the type of sugar, right? We said it's a deoxyribose. Okay, we're taking that, of course, from the deoxy. Uh, from the DNA, right? So which is, of course, deoxyribonucleic acid. Okay, so we're taking deoxyribose from there and then add sugar. So this is the sugar, and this is how you draw it, guys. It's a pentagon, right? That's how you draw it. And we said that each nucleotide also includes a phosphate group, right? So it also said we have a phosphate group and, of course, a particular nitrogenous base, right? So of course, for the DNA has um, 4 nitrogenous bases, which are your thymine. Okay, I need you to note all of these: thymine, adenine, guanine, and cytosine. And we said that we can maybe memorize that by, of course, this, which is at GC. The at, of course, is ANT. So we know that adenine always bonds with thymine, and guanine always bonds with cytosine. Okay, so that's what we were saying. Okay, so describing the structure and its functions. Okay, right. So now, how is the RNA different from the DNA? Because they like to ask that question. Okay, and yeah, it is examinable. You have to be able to differentiate between the two and see similarities. That will help you, of course, memorize everything that you need to know. Okay, so of course, the RNA is a nucleic acid, but its its acronym is actually ribonucleic acid. That's what it means, ribonucleic acid. Whereas the DNA is deoxyribonucleic acid. Okay, so I hope you got that. Okay, so that is what RNA stands for. And the first thing that we note that is different uh, when you're considering DNA and RNA is that for the RNA is single-stranded. Okay, so what do we mean by that? That we mean that it actually has only one strand. Okay, unlike DNA, which has 2 strands, the RNA has one strand like that. Okay, so it doesn't have a second strand that it is bonded to, um, unlike the DNA. Okay, so so we're saying that that's one difference. Okay, so we note that. Okay, I've highlighted this. Okay, the DNA is double-stranded, the RNA is single-stranded. Okay, and the RNA is also made up of nucleotides as well. Okay, so it also has this structure. Its nucleotides are like this, but of course we will explore the different types of RNA. Okay, so we're saying that RNA nucleotides are made up of a ribose. Okay, a ribose sugar. Now that's a second. Okay, so the first difference is that the DNA is double-stranded. Okay, and the RNA is single-stranded. Okay, second difference is that the DNA has this type of sugar, which is the deoxyribose sugar, and then of course the RNA has a ribose sugar. Okay, so that's the second difference there. Okay, need you to note those differences. Okay, so we have 2 differences so far. Okay, so we're saying that nucleotides of the RNA, particularly the mRNA, we'll get to that. So we're saying that each nucleotide is made up of a ribose sugar, a phosphate group, and a nitrogenous base. Okay, so a phosphate group and a nitrogenous base. So of course, both of these two are similar because the DNA has those as well, particularly the phosphate group. Now, the third difference that we have is that the RNA, instead of having thymine as its nitrogenous base, as one of its nitrogenous bases, it actually has uracil as one of it. So uracil replaces thymine. We don't have thymine in RNA, we have uracil. So that's a third difference there. Okay, DNA has thymine as its nitrogenous base, while RNA has uracil as its nitrogenous base. Okay, replacing thymine. And of course, the others are the same: adenine, guanine, and cytosine. Okay, so note those differences, guys. Okay, right. Now, RNA plays a huge role in protein synthesis, as we shall explore that. And so this, of course, represents the structure of the RNA. Of course, there we have the phosphate group, and there is the ribose sugar, right? The ribose sugar, and of course, this is a nitrogenous base. Okay, Right now, let's look at this. We actually have two types of RNA. Okay, we have what we call the messenger RNA. Okay, and we have an abbreviation there of mRNA. So we call it the mRNA, but it's really the messenger RNA in full. And it is actually formed. Okay, so it's not found anywhere, unlike others like your DNA. Remember DNA, we said that you have DNA, um, you have DNA inside the nucleus, right? Which we call the nuclear DNA. You have DNA um, in the mitochondrion, which we call empty DNA or mitochondrial DNA. Okay, so this applies for both animal cells and, of course, plant cells. Okay, so both of these two, so you have both nuclear DNA and mitochondrial DNA in both animal and plant cells. But the third type of DNA that you find, you'll only find it in plant cells, which is what we call the chloroplastic DNA. Okay, so it's the chloroplastic DNA. So we find it only in plant cells. So we said that these are the types of DNA that we have. Now for the RNA, we're saying that you don't find these, unlike the DNAs where you find them in these particular areas. For the mRNA, it's actually formed. Okay, it's made before it exists, so it is made inside the nucleus. Okay, during protein synthesis, and we will start with that. So during the transcription stage. Okay, so it's formed in the nucleus, and what happens, and it attaches on ribosomes. Okay, we will explore that. That is part of the protein synthesis. Okay, so that is your mRNA, and the mRNA looks like that. Okay, it's saying that we said that it is single-stranded, and of course it has these nitrogenous bases and ribose sugars and phosphate groups, which make up, of course, a nucleotide. So that's one nucleotide. That's the second. That's the third. That's four. So that's four nucleotides, making up that backbone of that mRNA, right? Okay. And then the second type of RNA that we have, we say that it is the tRNA or the transfer RNA. Okay. So unlike the mRNA, it's not formed during your protein synthesis, but it is found in the cytoplasm. Okay, so you'll find it in the cytoplasm. Remember, um, cytoplasm, we said, is a jelly-like substance that, of course, suspends all of these organelles inside the cell. Okay, so this is your cytoplasm. Some people say that it is actually just empty space, which is really not true. It's a jelly-like substance, and inside, we said that inside, if you really watch um, my my YouTube videos, you realize that actually we also have another jelly-like substance inside, which we call the nucleoplasm, right? So we call that the nucleoplasm. I should have maybe written that here. So so that is also a jelly-like substance, but it is inside the nucleus, right? So both of these are jelly-like substances. Okay, inside this is inside the nucleus, and that is inside the cell, okay, as a whole, right? So we're saying that tRNA, we find it in the cytoplasm. It has this clover leaf structure. You can say it's a hairpin structure, looks like that. Really hope that, yeah, it's actually a proper drawing. So you would find that each tRNA has a place of attachment for amino acid. Okay, so this is an amino acid, right? So it has an amino acid that is attached to it. And when you take a group of amino acids, okay, so this is what I'm drawing, a group of amino acids. It could be, I don't know how many, and they bond together. by peptide bonds. So this is so you will form what we call a protein, right? So a protein is actually formed by or it has monomers that we call amino acids. So many amino acids, they make up one protein, so on and so on. Okay, so that's why we need these amino acids so that they can bond with other amino acids, and that combination forms a molecule, a protein. Okay, now let's move on then, guys. Okay, right. So of course, as you can see here, this tRNA has space for actually three nitrogenous bases. Okay, three nitrogenous bases. So you'd find out that could be written adenine, uracil, and guanine. Okay, so these triplets, okay, this whole triplet, like all of these, is we give it a term. Okay, so this group of nitrogenous bases found in the tRNA, we call that group an anticodon. Okay, anticodon. And if you um, happen to forget, remember this is tRNA, right? So tRNA, of course, tRNA will be. So if you are wondering which is associated with which, because the mRNA, it's so for the mRNA, these three pairs, okay, so these three pairs of nitrogenous bases, for the mRNA, we call them codons. Okay, so this is a codon. It's an arrangement or a group of three nitrogenous bases in an mRNA, right? So we give it a term, we say that it is a codon. Okay, but for the tRNA, these three or these triplets or this group of three nitrogenous bases, we call them an anti or anticodon. Okay, so if you happen to maybe confuse the two, which one is associated with which, just look at the T's. Okay, tRNA, anticodon. Okay, so I hope maybe it does sort of help you in remembering, because you can struggle with remembering terms. So these guys, everything that I've just mentioned is very important. So I need you to really study that, take it into heart, and you will get full marks. I'm telling you. Okay. Right, so let's move on then. Okay, so that is your RNA, guys. That is everything you need to know. The location, okay, know where mRNA is formed, know where tRNA is found, okay, and know which, of course, which codons or anticodons are associated with what RNA. Know its structure, okay, the structure of the RNA itself and the structure of its nucleotides. Okay, note the differences between DNA and RNA. Okay, right. So let's then, we said that RNA plays a role in protein synthesis, and we also said that DNA actually provides a code that will initiate or start protein synthesis. So both of these play a role in protein synthesis. Okay, and what's the second function of the DNA? We said that its second function is that it carries genetic information to carry hereditary information from um, one generation to another. Okay, so it is what allows us to, or what causes us to look alike, like our family members, our parents, so on and so on. Okay, so now let's look at this protein synthesis. Of course, the word, as the word suggests, synthesis. When you're synthesizing something, you are basically making it. Okay, so you are making it. So what are we making here? We are making a protein. Okay, we are making proteins here. Okay. Now, for the protein synthesis, you need to be able to describe it, guys. Okay, you need to know the sequence of events. Very important. Okay, it's even mentioned here. It's even mentioned here, guys. Okay, we've already covered, of course, the types and location of RNA, the structure of RNA. Now, this is what we are going to cover. Okay, all of this, right? So, and so it's even written sequence of events. Okay, so we need to go through that. It's very important that you know the sequence of events, even in your DNA replication, which is quite similar to the protein synthesis. Okay, when it begins. Okay, so the protein synthesis is made up of two stages, right? The first is the transcription. Okay, transcription. This is taken from scribes. Okay, so back then there were no photocopy machines, and so there were people that would copy written whatever written information. Okay, so they would copy that information. So, and you will see how this really relates to this analogy. Okay, so that's the first phase or the first stage, transcription, and it occurs in the nucleus, inside the nucleus. And this is why I have this nuclear membrane, right? So this is nuclear, as the nuclear membrane, inside of course is the nucleus, just like what you see here. So it's actually, as you can see, there we go, that's the nuclear membrane, and inside is your nucleus, right? So let's take a look at this. What happens? So we have transcription. So in transcription, the first thing that occurs, or the first three steps, really look like or are exactly the same as DNA replication. Because what happens is that this DNA, we have a DNA, right? So DNA, we know that it has this double helix, right? Okay, has this double helix, and this double helix, the first thing that happens is that it unwinds. Okay, by unwinding, guys, we basically meaning that it untwists. Okay, it untwists. Remember, it is a twisted ladder like that. Okay, twisted ladder like that, and so it goes from this structure to side by side like that, like a ladder. Um, okay, like that. So that is what we mean by unwinding. Okay, so it goes from being twisted into being untwisted. So that's the first steps. Okay, say that the double helix DNA unwinds, right? The second step is that it unzips. Okay, so the double-stranded DNA unzips, and in the previous uh, lesson, we said that by unzipping, we mean that its weak hydrogen bonds between the complementary nitrogenous bases actually break. Okay, that's what we mean by unzipping. Okay, so that's the second step, that it unzips, or you can say that weak hydrogen bonds break. Okay, third step, the two separate strands are formed. Okay, so now you go from having a structure that or DNA that is untwisted, that has its weak hydrogen bonds broken, and so of course it will look something like this. Okay, so you have two strands separated like that. Okay, and in the DNA replication, we said that both of these act as a template. Okay, um, but for protein synthesis, what actually happens is that one strand is used as a template. Okay, so not both of these. So one of these will act as a template. The other will not be involved. Okay, so we are only using one strand in protein synthesis to form the mRNA. That's the goal. We want to form the mRNA in the nucleus. Okay, and so the mRNA is complementary to the DNA. Okay, or we can say that free nucleotides that are inside this nucleus in the nucleoplasm. Okay, so those nucleotides that are in this nucleoplasm, they bond there to form the mRNA, okay, the strand. Okay, so inside this nucleus, you actually have a nucleoplasm, and you have free flowing nucleotides. Okay, I drew them in the cytoplasm, which is of course incorrect. So they should have been inside, right? So let's start then building up our strand, the mRNA, using this um, single strand of the DNA. So for thymine, we said that thymine always bonds with adenine. Okay, so we'll have a nucleotide that is inside this nucleus, of course, bonding with that thymine. And then cytosine always bonds with guanine, right? So, and then we go on and have a nucleotide with the guanine nitrogenous base bonding there with that particular nucleotide. So, and then this will be guanine as well. The adenine, now this is where the problem starts. The adenine, guys, for the mRNA, because we are building an mRNA, we cannot say that adenine will bond with thymine. But of course, because the mRNA replaces thymine with uracil, and so that means that we're going to have a uracil nucleotide there. Then an adenine again there, and then of course we have a cytosine. Okay, so this is our cytosine. So in this way, we have created rather use white. We have created this strand, okay, the mRNA strand. We have created the mRNA actually. So there we go. That is your mRNA, guys. Now the next step, because now we have created that, now the next step is that it goes through the nucleopores, and it then, of course, waits or sits on the ribosomes. Okay, so this means that we will have this. Okay, let me rather use a thicker line. So, okay, 2, 3. So now we have, so it's AGG. So AGGUAC. Okay, so let's copy that down. It's AGGUAC. Okay, hope you do see this is the mRNA, right? So it actually attaches onto the ribosomes like that, okay? And it has that code. So after you have so that's the last step, right? When you have your mRNA exiting the nucleus through the nucleopores, and then attaching onto the ribosome. That's the last step of the first stage, which is the transcription. Okay, so the mRNA, we say that it has a coded message for protein synthesis, and then it moves, okay, from the nucleus to the cytoplasm, and then attaches to the ribosome. Okay, so that's what we're saying there. So that is the whole transcription. That's the first stage. Okay. Now, what's important is that you note that it actually carries this code, and you are able to mention or identify the complementary nucleotides, or rather nitrogenous bases, that will bond, of course, with the DNA, because the thymine part tends to really confuse students. Okay, Now, what is happening now on the second stage, which is the translation, actually happens in the cytoplasm, right? So the first step is that each tRNA carries a specific amino acid. There is your tRNA in the cytoplasm. There we go. Okay, so we have each tRNA in the cytoplasm like that. Okay, and of course it's carrying um, um, with it along an amino acid. Okay, which will bond with other amino acids to form that protein that we need. And then the second thing that happens is that the anticodon on the tRNA matches the codon on the mRNA. Now let's look at this on this diagram. So saying that um, we have this group. Okay, I'd rather use colors, guys. Said A. Okay, let me rather use yellow for A, and we used okay, G. Okay, so these three, this set, like where you have A, G, G. What is that? That is on the that is a triplet, right? On which type of molecule? We have it on the mRNA. So what do we call this then? We call this the codon, right? Because on the mRNA, call it a codon. Okay, and then we have another set again, another triplet, which we also call it a codon. Okay, so each triplet here, each group of three nitrogenous bases, we call them a codon, especially when they are on, only when they are on an mRNA, right? And then of course with the tRNA, okay, so look at that in scale. tRNA, this triplet we call an anticodon. Okay, so that is an anticodon. Now each triplet will match with, of course, each codon. Okay, so this is UCC. UCC, it already has its respective amino acid. So UCC is going to, of course, move towards the uh, the mRNA, and of course UCC means that it will bond, it will match with this first triplet. So that's UCC, right? And of course its having its um, amino acid, its unique amino acid attached to that. Okay, so as you can see, we have actually matched the anticodon, okay, with the codon. Okay, I mean the codon with the anticodon. Okay, so there we go, there we have. So that's what you are having there. And then let's look for another tRNA so that it matches with this codon. This is UAC. So it should be AUG, should be. So it should actually move to that direction. So it moves and attaches, okay, proper way. So to attach that way, in that manner. Okay, that is U. And so this should be A. Okay, that's A. This should be U, and that's C. This should be G. Okay, so be careful of the confusion we usually have uh, between thymine and uracil in these two different molecules. Okay, so that's what we're doing there. And so what happens after the matching? So tRNA brings the required amino acid to the ribosome, of course, and then amino acids become attached by peptide bonds to form the required protein. Okay, so I like the second stage. It's really short. So of course, this will happen on multiple with multiple codons and anticodons. Okay, so it won't be just two as in this case. So we're saying that these amino acids will be bonded together by what we call a strong peptide bond. Okay, that is what you need to know. It's a peptide bond, guys. It's between consecutive amino acids. Okay, and of course these many of these. Okay, so you have plenty of amino acids having um, being bonded, of course, by this type of bond, which we call the peptide bond. Okay, so that will then we said that multiple amino acids make up a protein, right? So they make up a protein, and so that means that this is a protein. Okay, this whole these multiple amino acids form one protein molecule. Okay, like that, and that's it, guys. Okay, so um, let's note everything, okay, and let's not rush when we are writing down our answers during tests, during exams. Let's go step by step. Okay, that's what life sciences need. Okay, and the terminology is very important. You cannot use like each thing carries a specific. You cannot use those words. You have to use scientific words. Okay, so that's how we test you. Right, we're done um, with the content, with the breakdown. Okay, so now what we have to do is to finish this up with activities. Okay, let's start with the first activity. Okay, so this looks sorry about this. This looks like give one word. Yes, it is actually. Give one word. So this is where you should score your marks, guys. This is one of the easiest, especially if you know that you study most of the time. Sorry about this. Right, so let's go. So give the biological term for each of the following descriptions. Right, okay, let's go. The sugar, so the first question says the sugar found in RNA, what do we call that? We call that the ribose sugar. It's different from, of course, the DNA sugar, which is the deoxyribose sugar. Second question, the bond that forms between two amino acids. We just described that now, explained that. So we call that the peptide bond. Okay, let's move on. Number three says openings in the nuclear membrane that allow the mRNA to leave the nucleus. We said those are the nuclear pores. Pause. Right. Then question number four. The stage of protein synthesis during which mRNA forms from DNA. That's the first stage, guys. That's where we are forming mRNA. We said that is the transcription. Okay, transcription. I hope you did find some connection between the analogy I was making and the phase, of course. Number five says the type of RNA containing anticodons. Anti T, so that's tRNA. Okay. Um, Number 6, okay, says the organelle in a cell where translation occurs. The organelle in a cell where translation occurs. Well, it said that it occurs on the ribosomes, guys. Occurs on the ribosomes. Number 7, the organelle in the cytoplasm, which is the site of protein synthesis. Okay, so if this is the organelle in a cell where translation occurs, then that should be that should be the cytoplasm then, where translation occurs. Yeah, yes, yeah, yep. I made a mistake there. And then the organelle in the cytoplasm, which is the site of protein synthesis, that is your ribosome. Okay, and then number 8, the type of nucleic acid that carries a specific amino acid. Come on, guys. What type of nucleic acid? That is RNA. Don't say tRNA. Just talking about types of nucleic acids there. So, or let's rather go with tRNA. I think, yeah, tRNA. Unless it's a trick question. And then number 9, nitrogenous base found only in RNA molecules. That is uracil. Okay, hope that we got that correct. Let's move on to the second activity. Okay, um, so it says the diagram below shows part of a process involved in the production of a protein. Okay, so start with 2.1. What do we see first? Well, this actually looks like the stage where they are matching. So this is the translation stage in the cytoplasm on the ribosome. There is your ribosome there. Okay? This structure. Okay, they like to draw it like that. Okay, so 2.1 says identify molecule Y. Molecule Y, this molecule over here, and of course that is the tRNA, guys. That's the tRNA, right? That's 2.1. Then 2.2 says identify the group of nitrogenous bases Z. So this looks like we have this, right? So this tRNA looks like it will attach onto this next, okay? Because remember, this peptide bond has to continue on, so this will connect there, and so this means this is the codon, okay, on this mRNA. So we're looking for the anticodon, okay, or the anticodon there. So this is ACC. So, and we are going from left to right like that. Okay, that's 1, all the way to 3 to the right. So that means that we're going to have, so for A, it's going to be U, for C, that's G, and for another C, that's G. Okay, so we're going with ACC, right? Okay, then for 2.3, if X is the next amino acid required after W, identify nitrogenous bases 1, 2, and 3. Okay, so I made a mistake here. So I guess this should, okay, so I already have this covered. So the group of nitrogenous bases Z, guys. Okay, I made a mistake there. Okay, so do you see how important it is to read the question? So really important. Sorry about that. So we're actually referring to this group. So this, what do we call this group of nitrogenous bases in this tRNA? Of course, we call those the anticodon. So that is the anticodon, and yeah, attaching onto the codon. And then for 2.3a, we've already tackled that question, answered that question. So let's move on to 2.3b. Okay, the DNA base triplet that codes for X. The DNA base triplet that codes for X. Okay. Okay. So it has to be DNA, guys. That is coding for X. Okay. So first of all, X attaches. X is an amino acid, which of course is attached to this tRNA, which has this anticodon, which is ACC, right? UGG. Oh, sorry about that, guys. I keep on making a lot of mistakes here. I don't know what's happening. It's supposed to be UGG, right? Okay. So for X, we said that it is UGG, and so DNA that codes for this, DNA-based triplet that codes for this, of course, will be what? ACC, right? But that is, of course, what looks like. I think what they are trying to ask is, what is the complementary triplet for this mRNA right here? Okay. So because it's ACC, then we'll go with TGG. Okay, ACC-TGG. That's how this mRNA was made. This part of this mRNA was made. We had a TGG triplet in the DNA, and then of course its complementary codon is that ACC, and the complementary anticodon is that UGG. Okay, hope that's correct. I really hope so, guys. If not, it's going to have to be C. It has to be ACC. Yeah. Okay, let's move on to. Oh, actually, it's number 3. So describe the process of transcription. Of course, guys, you begin um, your description with the DNA replication part, the unwinding, then it unzips, and then 2 separate strands are formed. One strand is used as a template to form the mRNA, and then the mRNA is complementary to the DNA, and then it receives the message and it moves out of the nucleus. Okay, so you are going to write that down. I'm just thinking that these questions can really be tricky sometimes. Yes, they can really be tricky, as you saw for yourself. Okay, so that's what, of course, you need to write down. Okay, try to ensure that you write down these words. They're very important words. They're looking for these words, and don't rush. Try to describe every steps. Okay, describe every single step and in order. Okay, they also look for that. Right, let's look at the third activity. The sequence of amino acids in a protein molecule is coded for by the DNA and RNA. The table below shows some mRNA codons and the corresponding amino acids. So these are the mRNA codons and their corresponding amino acids that are attached to the complementary tRNAs that are going to match, of course, with these codons. Okay, so let's look at 3.1 then. 3.1 says, according to the table, how many codons code for phenyl phenylalanine. I hope I pronounced that correct. Um, so how many codons? Okay. Well, we have how many codons here? That's one codon for that amino acid. This is the second codon, right? So should be two codons, right? Codons, yes, two codons. Let's move on to the second questions. Uh, so the second question is 3.2, and it says, what is the anticodon for glutamic acid? Where's glutamic acid? There it is. Okay, so we're looking for the anticodon for this. So because this is GAG, the anticodon of this would be CUC. Okay, CUC. Remember, A for RN for all RNAs, it's always going to bond with uracil. Okay, replacing that thymine. They want you to make that mistake. Don't fall for it, guys. I'm on your side. Um, should have been 3.2, right? Yeah. Then 3.3 says, a section of mRNA has the following base sequence. Okay, so it's a section of mRNA, and it is read from left to right. Okay. A says, give the DNA base triplet for the last codon on this section of mRNA. The last has to be with the one on the right because the sequences, remember, this is read from left to right. So we're looking for a DNA triplet, okay, that is complementary to this. ACC, so ACC will have to be TGG, right? TGG, thymine bonds with adenine. This cytosine bonds with guanine, both of those, okay? So this should be A, right? Okay. The way I'm making mistakes today. And then B says, give the first amino acid coded for by this section of mRNA. The first amino acid. So the first amino acid has to, of course, be GAU. GAU. There we go. Okay. Aspartate. Okay. So that's what we write down. Okay. This is just using, of course, the table here. And then 3.4 says, name and describe the process occurring in the nucleus which results in the formation of a mRNA molecule. First of all, the first thing that you should do is to name. Okay, you have to name it. So what is that stage? It's transcription, right? It's the transcription part of the protein synthesis. Okay, and then you describe it. Okay, I described it in this previous activity. So, of course, you know how it goes. Let's move on to the next one, the last one. This is the last one before we wrap up this session. 8.1 says, state two differences um, between a DNA nucleotide and an RNA nucleotide. We went through these, guys. What did we say? Said that maybe let's tabulate. Okay, tabulate there, and then RNA. So the first things first is that the DNA has thymine instead of uracil, right? Okay, has thymine instead of uracil as as a of course a nitrogenous base. Okay, whereas RNA has of course the opposite, which is uracil. So uracil replaces thymine. Okay. Okay, let me just write it of thymine. Okay, right, that's the first step. And the second difference is that this DNA has a deoxyribose sugar. Remember, we're talking about their respective nucleotides. Okay, so this has a deoxyribose sugar, and the RNA has a ribose sugar, guys. Okay, can't go wrong there, right? Okay, so um, this should be it, guys, for our lesson today. I'd like you guys again to please share this, send it to friends, subscribe, like, and comment. Let's interact here, guys. I need more interaction. You guys have been quiet. You guys have been quiet. Come January, everyone will be here. Okay, so please let's start this now. Let's start growing this channel, as it will help a lot of people and it will help, of course, achieve goals. um, and so I really would like for this channel to grow. We are close to 50 subscribers now. We still need more. Let's aim for 100, guys. We want 100 by the end of January. So let's aim for that. Okay, so I'll see you guys in the next lesson, and have a good day.